Welcome to UMDF's Ask the Mito Doc monthly webcast. Tonight we will hear two perspectives on POLG. Dr. Bill Copeland from the National Institute of Environmental Health Sciences will share insights on POLG and his research findings over the years, and then followed by Dr. Bruce Cohen from Akron Children's Hospital, expanding on the clinical side of POLG. Moderating this evening is our very own Margaret Moore. Our Ask the Mito Doc webcasts are made possible through the generosity of sponsors, so we ask that you join us in thanking the Edith Trees Charitable Trust and UCB. The holiday season is upon us. Please check out the beautiful stories behind this year's Give Energy. Donate and/or share to help us continue programs like Ask the Mito Doc. If you or a loved one has been diagnosed with knee loss, our friends at Sacenta Therapeutics would love to hear your patient journey. Please visit umdf.org/studies and surveys for details. We are already busy making plans for our next UMDF annual conference for patients, families, researchers, clinicians, and industry. Meet us in St. Louis for Mitochondrial Medicine 2025, June 18th through the 21st. Save the dates now and more details to come at umdfconference.org. Please note, the information provided in this webcast is for general information only and should not be relied upon as a substitute for professional consultation with a qualified healthcare provider familiar with your individual medical needs. Please consult your primary physician and/or specialist before changing the course of your care.
Hello, everyone, and welcome to our November Ask the Mito Doc. At this time, I'm going to invite Dr. William Copeland from the NIEHS to join us on screen, and we're going to get his presentation loaded and get ready to go. Dr. Copeland's going to talk about POLG and health and disease tonight. And after he's done, Dr. Bruce Cohen will pop on.
Thank you, Margaret. Okay, thank you for the introduction. Get a laser pointer. All right, we'll go ahead and get started. So I've been asked to speak a little bit from the research aspect about POL gamma or POLG and its role in health and disease and what we've learned from a research perspective. So let me get right into it. So this is a little bit of the outline. I just want to first introduce the mitochondrial DNA because that is actually the substrate for the POL gamma or POLG, and because its role is to copy that mitochondrial DNA. And what I want to kind of describe is how mutations arise in that mitochondrial DNA and the role of POL gamma in presenting or actually in health and disease and in a disease state, and how it can cause more mutations, and in a natural state where it actually can prevent some mutations. And I kind of want to end with some exciting stuff about therapies that are on the horizon. All right, so as many of you know, our mitochondria have their own DNA, and it is much different than the DNA in the nucleus. It's a circular genome. It's actually very small compared to the nuclear genome. And I've got a little outline here. It's about 16,569 base pairs, and it's circular. That's compared to our nuclear DNA, which is over 3 billion base pairs, and you have two copies, so you actually have about 6 billion base pairs of that. In terms of the genetics, it's very distinct from our nuclear DNA because we know it is maternally inherited. And again, unlike nuclear DNA, where you only have two copies, you can actually have thousands of copies per cell. And that copy number can be very dynamic because of the energy needs of that tissue. And that allows it some buffering capacity, which I'll get into a little bit later. So the main function of our mitochondrial DNA is to encode. It has 13 genes that encodes for 13 proteins. We call these gene products. And those 13 proteins combined with nuclear encoded proteins to form the electron transport chain, which makes up and carries out oxidative phosphorylation. And so this is a dual sort of process that requires proteins from both the nucleus and the mitochondrial, and they have to combine and match to carry out that function. What we do know from the last 40 years is that mutations can arise in this mitochondrial genome, and those mutations can be in the form of point mutations, where like you'll remember from your biology, like A has to match with T, G has to match with C. Sometimes there's mutations that prevent that. and you get mistakes within this DNA. You can also get deletions. So you can get large chunks of this genome can actually be chopped out, and those are called deletions. And then another disorder we would call depletion, where you just basically lose a lot of the DNA. So all these give rise to a slew of a subset of diseases, all coming under the umbrella of mitochondrial disease. But because of that high copy number, there represents an issue, unlike in a nuclear genome, where you can get mixtures of both wild type and mutant copies, and that's better expressed here. So, the best case scenario, your cell would have all wild type copies. But sometimes, and it's very common, you can have mutant mitochondrial DNA. And depending upon the level of that mutant mitochondrial DNA, if you get above a certain level, it'll present with a disease state. And we know that level is well above 50%. In other words, your cells have to contain more than 50% mutant mitochondrial DNA before that critical threshold is surpassed, and where you get a disease state. But this large copy number offers a little bit of buffering in terms of allowing for a large amount of some mutations. Of course, those mutations give rise to disease states, and those can affect multiple organs. And one of the hallmarks of mitochondrial diseases is that it usually affects more than one organ at a time. So I want to speak how the mitochondrial DNA gets replicated. So what we've learned from both a nuclear and mitochondrial DNA is that DNA is copied by what enzymes we call DNA polymerases. And basically the DNA polymerase is there to match. It reads at the template DNA, and it has to, across a certain nucleotide, so for example, across an adenine, it's got to match with a thymine. Likewise, if it's copying a cytosine, it's got to put in a guanine. And that allows it to what we call undertake high fidelity DNA replication, because we don't want this enzyme to make a lot of mistakes. So in human cells, there's actually 17 DNA polymerases, so there's quite a lot. But the only one for this audience and for what I study, the only one that we really care about is the POLG gamma, which is encoded by the gene POLG. And that's because of all these 17 DNA polymerases, it's the only one, POLG gamma, is responsible for replicating our mitochondrial DNA. All these others are involved mostly in nuclear DNA replication or nuclear DNA repair. And for that reason, especially in my group, we focus a lot of attention on the POLG gamma. This is a cartoon of a replication intermediate, and I'm introducing all the players that we study. So the POLG gamma is actually a heterotrimeric complex. That means it's composed of 3 proteins. The main protein is this POLG gamma. It is the DNA polymerase, but it also has what's called an exonuclease function. It combines with a dimeric accessory subunit, and that helps keep it on the DNA. DNA has to be unwound, and that's afforded by this helicase, which we call twinkle. And that generates also single-strand DNA, which can be attacked by certain, it makes it very vulnerable, but that's protected by this single-stranded DNA binding protein. Now, what we know about this mitochondrial DNA from the history is that it mutates or evolves much faster than the nuclear DNA. And what we've been trying to understand in my group is why that's happening. And so when we look at the fidelity of DNA replication, we can pretty much figure out why. So if you look at nuclear DNA replication, it replicates with making less than 1 error per every 10 to the 10th bases copied. And that's afforded by about 100,000, about 10 to the 5th insertion fidelity by the DNA polymerase. That's the matching of those bases. And that's equivalent to if you're using a word processor, if you're typing. And as if you know, when you're typing, you often make mistakes, and the first thing you want to do is hit your backspace or delete. That is equivalent to the enzyme proofreading. So many of these polymerases have an exonuclease function that they can catch that mistake. After the polymerase has actually copied the DNA, in the nucleus at least, any further copies can be error checked by what's called DNA mismatch repair. And that's equivalent to, like, after you're done typing, you turn on your spell check. The problem is in the mitochondrial is that there is no mismatch repair. In fact, the mitochondria actually have a limited amount of repair pathways as opposed to the nucleus. So it really only has one efficient repair process, that's this base excision repair, but it lacks mismatch repair. And for that reason, we believe that the major driver of mutations, because of that lack of mismatch repair, is actually spontaneous errors of DNA replication from Paul gamma. And this is the normal process of how mitochondrial DNA gets replicated. But I will say this enzyme is a high fidelity enzyme, and it only makes about a mistake about 1 in every million bases copied. Nonetheless, that's enough to cause a low level of mutation. So by the time an old person in their 90s or so would have still less than 1 or 2% mitochondrial genomes that have carried these mutations. So in a healthy individual, this is really insignificant. However, in the disease state, that's a different scenario. So I introduced these 4 players. The problem is all 4 of these genes for these proteins have now been identified in the clinic as disease alleles that when mutated will give rise to a mitochondrial disease. And those are some of those diseases are shown here. Mutations in the Paul gamma can cause these diseases, starting with early childhood Alpers-Huttenlocher syndrome, all the way to some of the later onset ones, progressive external ophthalmoplegia. Likewise, this PEO or progressive external ophthalmoplegia can be caused by POLG, POLG2, as well as a twinkle. And one of the newer ones that was discovered about 5 years ago was mutations in this gene, and that causes optic atrophy. Now, the big player in these diseases, of course, is Paul gamma. And that's because there's well over 300, upwards of 350 disease-causing pathogenic mutations that have been identified in this gene that give rise to these substitutions. So what I've diagrammed here are the single letter amino acid substitutions from those disease mutations in the gene. And they're color coded by the disease state. So the ones in red are causing an early childhood infantile hepatocerebral syndrome or Alpers syndrome. That's a recessive disease that causes loss of your mitochondrial DNA or mitochondrial DNA depletion. The ones in black are associated with ataxia neuropathy. Unlike the early childhood ones, that doesn't cause depletion, but it causes deletions within our mitochondrial genome. And that's also recessive mutations. Down here are various forms of that progressive external ophthalmoplegia. Most of these are recessive. They also, like the latter diseases of Paul gamma, they cause deletions in our mitochondrial genome. There's also a cluster of dominant ones that we know that there's 2 genes for everything in the nucleus. So in a dominant case, you only have to have one of these mutations to get a disease state. So I know Bruce is going to cover this in more detail. Basically, a synopsis of Paul gamma diseases, the early childhood ones, either mild cerebral hepatopathy spectrum disorder or Alpers-Huttenlocher, they cause depletion of your mitochondrial DNA, and they usually present very early, 1 to 2 years of life. Later in life, starting with the teens, ataxia neuropathy, myoclonus epilepsy, and the progressive external ophthalmoplegia, they're all associated with deletions. So it turns out that Paul gamma mutations are some of the most common causes of inherited mitochondrial disorders. And it's been estimated that approximately 2% of the general population is carrying a pathogenic genetic variant of POLG. Fortunately, most of those are recessive, and in other words, you're just carriers. But occasionally, if 2 parents are both carrying a recessive mutation and they pass both of the bad copies on to their child, they can give rise to one of the Paul gamma diseases. And the overall prevalence of that is about 1 in 10,000. So one of the challenges we face in my group is what we've done over the last 25 years is study a lot of the consequences of these mutations and kind of understand the severity. But one of the challenges we've met with is that we've realized that just because a patient has a certain Paul gamma mutation, it doesn't mean they're going to have a specific disease. And an example of that, I'm going to give 2 short examples. This is an example, a chart of literature review of these Paul gamma variants. So this T251I P587L is a pair of allelic mutations, meaning they occur on the same Paul gamma copy or same Paul gamma gene. So that's what we call allele 1. And these patients, they also have a mutation on allele 2. And so you can see there are different genotypes. And what I want to illustrate here is I want to just zoom in on these 3 cases. So in these 3 patients, they have the same Paul gamma genotype. They have this allelic pair mutation on one allele and this G848S on another allele. But what I want to point to you is that the age of onset varies significantly. Here's a patient that developed at 6 months of age of the Alpers, which is the most severe disease, whereas these other 2 didn't present until 51 and 73 with PEO and Sandow. So there's a very a great variation in presentation with the same POLG genotype. Here's another situation. This is homozygous A467T patients. This is work done by Shamima Rahman in London. So she looked at, has 4 patients. They presented at 3, 6, 20, and 24. This patient died at 5.5 from seizures. This one's still alive at 16. This one died at 44. And this one's still alive at 31. So there seems to be a great variation. And that's kind of some of the complexities of trying to understand the Paul gamma diseases. So why is there such a variation? Well, the mutations in Paul gamma will cause defects in the enzyme, and that will cause the defects in the mitochondrial DNA. And so there's a chain of events that have to happen, but it is also influenced by many other things. It could be influenced by the rest of the genome, other nuclear DNA variants, or mitochondrial DNA variants. It can also be perturbed by environmental factors. We know that, I'm sure your clinicians of your family members have told you, please stay away from certain toxins like smoking and eat a healthy diet, and all those will help and prevent some of those environmental factors that can trigger and push the threshold of some of the disease over and where you actually will trigger the disease state. All right, so this is where it's really kind of a headache for us to try to understand the complexities of both nuclear mutations causing mitochondrial DNA mutations and being influenced by the environment. But we're still we're starting to get a handle on some of that. So I kind of want to go into some where we're going with all this and what does the future hold for the Paul gamma related disorders. So over the last 30 years, I've seen a great deal of increase in the appreciation of some mitochondrial diseases, and especially for Paul gamma related disorders. Right now, there's just for the clinician, there's lots of published literature, also for the families and patients to get a grasp of what's going on. We're now in a very readily available sequencing technology using either whole genome sequencing or whole exome sequencing. You can quickly identify Paul gamma mutations and also other associated mutations. And we have actually a website that's publicly available to help the clinician or the patient to understand the kind of the consequences of those mutations. And luckily, most of these the sequencing is covered by the health insurance in this day and age. All right. For the researcher, there are a lot on now more disease models to help develop therapies. We've had cell models for a while. There are now mouse models of Paul gamma, Paul G2, and Twinkle. We have developed one for the Paul G2, and some of our colleagues in Europe have developed ones for the Paul gamma. And there's a lot of good mouse models for that as such. There's also zebrafish models. The zebrafish is actually can be quite useful. It's very cheap to use compared to the animal models. And these are two. There's an example of a zebrafish model being recapitulating some of the Paul gamma disorders, and they're using that now to screen for drug treatments. And then there's organoids models. And one of our colleagues in Germany, this Christina Lang, has developed organoid models of Paul gamma directly from disease patients. And that is going to be very useful in work going forward to screen for some of some new therapies. And that kind of goes where this last kind of topic is therapies that are in the works. For a while, we've been working on antioxidant therapies, and that's still one of the main frontline drugs for POLG gamma mutations, as well as many other mitochondrial diseases. There's a lot of anti-seizure and anti-epileptic drugs. Past postdoc of mine, Shireen Chan, now has a company and using a zebrafish model has now developed drugs against epilepsy, and those are now are being reviewed by the FDA. There's nucleotide therapy, and you may have realized that nucleotide therapy has been incredibly useful for the diseases involving TK2, and that develops into Menji. And so the question has been, can it be applicable to POLG gamma diseases? And actually, many of our colleagues have already are using that in cell-based models. Here's this Christina Lang again, using some of her organoids models, and she's shown that in her system, at least if you challenge the mitochondrial DNA replication with this drug here, you can then rescue it by deoxynucleoside treatment. And so those are promising avenues as well. Metformin, which you've heard about, is kind of one of the most universal drugs, has actually been shown to ameliorate some of the defects shown again by Christina Lang with POLG gamma mutations in stem cell derived astrocytes. And finally, there are a lot of efforts for repurposing drugs and screening for new drugs. And there's two companies I know of working right now that are publicly have announced. There's this company, Stemnovate, is actually asking for volunteers that for clinical trials looking at Alpers-Huppenlacher syndrome. Pretzel Therapeutics is, along with some colleagues in Sweden, are developing directly for have a drug that are targeting some POLG gamma mutations. And my group is actually working with some Stanford University to develop some targeted drugs against specific POLG gamma mutations. So those are some of the things that are in the works to look for drugs. And those are just a few examples, but there's a lot of others. Of course, the gene therapy is probably the best, but we're not at a state yet where that's going to be really approved. But I think with that, I will stop and we can hand it over to Bruce. I think is that the goal now?
That's what we'll do. And if you could stop sharing, that would be great. And thank you, Dr. Copeland. It was so interesting. Every time I hear you speak, I learn something new, and those slides were excellent. So thank you for all of that.
Thank you. I'll look at the questions then.
Thanks, Bill. I learned a lot, and I left some questions for you. It's a pleasure to be here. I'm glad to see we've got a lot of people in the audience. I'm here to talk to you tonight about POLG disease. It's obviously a terrible illness, and I think we're all here dedicated to finding treatments and cures for this disease. So if I can have my first slide, please. Or my second slide. Okay, how about my third slide? So POLG is a rather a new term, and I'll get to the diseases itself in a little bit of time. But the real descriptions of the illness that we're talking about really date back almost 100 years. In 1931, Bernard Alpers coined the term Alpers disease to describe an illness that occurred in children generally between the ages of 3 and 7 with a terrible epilepsy. Most of these children died in the first year of developing their epilepsy. Some lived longer lives than that. But the illness was characterized by epilepsy, developmental regression, paralysis of the limbs, blindness, and then death. Science marched forward over the next few decades, but the next term that was introduced, or the next features of the disease that was introduced clinically, was introduced by Peter Huntlocker, who's a pediatric neurologist in Chicago that recently passed away, who described the liver component of Alpers disease. And when I was in training in the 80s, this illness was referred to as Alpers disease or Alpers syndrome or Alpers-Huntlocker syndrome or the Huntlocker variant of Alpers disease. And that's where we were really at until the late 1990s and the early 2000s. And I'll get to the features of these diseases shortly. The other thing I want to introduce is you'll see POLG written in regular straight type and in italic type. POLG is actually the name for the gene. And when we're talking about genes, the nomenclature is to put it in italics. That gene codes for the protein POL gamma or polymerase gamma. That's the protein that replicates mitochondrial DNA. So that's sometimes why you'll see the italics. And if you see it in straight print, we often use a straight print to refer to the disease itself. So I can have the next slide. Let's just go back one slide. There's a reference that I put here. We can go back one slide. POLG related disorders. This is a review that I wrote with Dr. Copeland and Dr. Chinnery. It was recently republished. I think this is our 4th edition of the publication. You can find it in Gene Reviews, the NCBI bookshelf. And if you just Google my name, Dr. Copeland's name, and POLG, this will be one of the first things that come up. And it contains evidence-based information about everything you want to know about POLG in its entirety. It's evidence-based, meaning that thoughts and opinions without backing it up with science don't end up in the final publication. So it's a bit dry reading, but it does have the information that you'll need. Next slide. Tara asked me to present a few cases, and I decided to really dig deep and go back. And the first patient I saw that I believe has POLG disease, I met in February of 1983. I had been a physician for less than a year. I was in my internship. And when you're an intern, you basically work inside the hospital system, not really knowing the patients for very long. This is a child that I took care of just for a few days. He was 7 years old. He had intractable seizures since the age of 3 years. He was started on an anti-seizure medicine called valproate 3 months before and had recently developed liver failure. This was manifested by both elevated liver enzymes, the enzyme we now know as ALT. The liver also makes albumin, which is the main protein in the blood. He had extremely low albumin, and the liver also makes the proteins that help blood coagulate. And when your blood when those enzymes aren't made, a test called INR elevates and results in spontaneous internal bleeding. I only knew this child for a couple days before he died of internal bleeding. Nowhere in his chart do I ever remember seeing Alpers disease or Alpers-Huntlocker syndrome, even though Huntlocker described it a few years earlier. But as I look back on this case now, I truly believe that he had POLG disorder. The next case, case 2, is a patient that I met as a senior resident in neurology. I had trained at Columbia Presbyterian, where there was always talk about Alpers disease. So when I saw the patient, I instantly knew what she had, although there was no genetic testing back then. And again, I believe this patient had Alpers disease or Alpers-Huntlocker syndrome or POLG disorder. I can't prove it. But I met her when she was 13 months old. She had a 1-month history of difficult to control epilepsy. Her initial brain CAT scan, that's an image of the brain, was totally normal looking. The child had normal development and behavior up until the first seizure. but behaved normally between the seizures initially. She was treated with phenobarbital, which was the most common anti-seizure medicine used back then. That did not work. Phenytoin, which was the second most common used medication back then, was added to the phenobarbital. That did not control the seizures. The third medicine was carbamazepine was added. That did not control the seizures. So a week prior to transfer to my care, valproate was added. The seizures actually improved with the addition of valproate. But over the next few days, the child became sleepy, less interactive, despite the fact the seizures were nearly gone. And a repeat CAT scan of the brain showed the entire brain had begun to shrink away from the skull. This is obviously a terrible consequence of the illness. And one day prior to the transfer, her ALT, that's her liver enzyme, was elevated to 500, which is about 10 times normal. So the doctors taking care of her knew she was in trouble. Next slide, please. What do we know about these two medical issues back then? Here's what we knew about Alpers disease. It's an epileptic encephalopathy. That means the seizures are unrelenting and the patient begins to drift away mentally, cognitively. This illness typically begins between the ages of 4 and 7 years, but it can begin a little earlier or a little later. Again, with progressive cognitive functioning dysfunction, blindness, and paralysis of the limbs. Long-term survivors may develop liver failure. That's what we knew back in the 80s and early 90s. I had mentioned valproate in case 2 and in case 1, and I'll be mentioning it again in case 3. This is what we knew about valproate-induced liver failure. And again, I want to make sure we realize that there was no link back in the 70s and 80s between these two entities of Alpers disease or valproate-induced liver failure. What we knew is that when we started patients on valproate, the incidence of liver failure, and it was fatal liver failure, was 1 in 50,000. Typically, the liver failure begins within 3 months of initiation of the valproate, and it was often seen in young children who had developmental delay or recent loss of developmental milestones, who had intractable, unrelenting seizures, and who had been using phenobarbital, phenytoin, and carbamazepine prior to the valproate. So what are we describing in valproate-induced liver failure? We're describing the typical child with Alpers disease, but we did not know that at the time. Next slide. So we're going to move on to case 3, and this was a career-changing patient that I saw. Let me tell you about her. She presented with her illness in 1993. I didn't meet her till 1998. But in 1993, she was a 15-year-old woman who was evaluated for jerky limb movements, and she was given the diagnosis of mannerisms, suggesting a psychological cause of the movement disorder. But that really wasn't the case. Next, if you can hit forward again. In 1997, at the age of 19, she was in college, and she had her first seizure. I'm not going to get into the details of the myoclonus, but she had her first seizure while in college. Her EEG, that's the brainwave test, showed that the seizures were coming from different parts of her brain. Her brain MRI scan, which is a more sophisticated image than a CAT scan will give you, was perfectly normal. And she was started on carbamazepine. That's the third drug that we were talking about before. This carbamazepine was ineffective, as would be predicted in a child with multifocal spikes on EEG. Her doctor changed her to valproate, again, noting that there was that 1 in 50,000 chance of liver failure, and the valproate stopped her seizures temporarily. Next slide. 3 months after starting the valproate, she was in florid liver failure, and despite the fact she was in bad liver failure, she was healthy enough to undergo a liver transplant. That liver is still working today, by the way. Next slide. So one of her doctors, and again, I was not taking care of her at the time, one of her doctors suggested that this could be a mitochondrial disease, suggested a muscle biopsy and a mitochondrial evaluation, and that was done. And without getting into the details of what was found biochemically or genetically, biochemically, something was identified that was consistent with the mitochondrial disease. Genetically, she had the best genetic workup you could have in 1998, and nothing was found. Again, we'll find out that POLG had been identified in 1996, but any diseases associated with POLG mutations had not been identified at this time. She was started on vitamins. Next slide. I first met her within a year of her liver transplant. In my first office visit, I noted that she had seizures that were poorly controlled. She had these jerky movements that appeared like myoclonus or another type of movement called chorea, which is another type of jerky movement. I certainly didn't think these were mannerisms. She was bright, energetic, and had returned to college and part-time employment. Next slide, please. I saw her again in 2000 and 2002. She didn't live where I was working, so she would travel every couple years to see me. There was no change in any of her features of her illness or her condition. She returned to see me in 2005, and things had begun to change. Next slide. There was some diagnosis in sight. So she developed some new symptoms and signs. If you can hit the return, advance forward. Her seizures for some reason were better. She developed droopy eyelids. She developed slurred speech. She was ataxic, meaning that she had very poor balance and coordination. She had very poor night vision. Her retinas had begun to deteriorate. She had developed hearing loss and lost her reflexes. If you can hit forward again. She had developed some memory difficulties. Next. She was no longer able to work at that time. And at that time, I actually, and that's her loss of reflexes. At that time, I actually, in my heart, knew what she had because I was reading the literature and what was going on in the field of mitochondrial disease. Next slide. So just as obviously I'm telling you this story because she had POLG disease. I last saw her in person in 2015. I saw her on telehealth during the pandemic, and because she lives in a distant city, she transferred her care to another mitochondrial doctor, and last contact with her was a few months ago. So we can go forward again. So this disease was originally called Alpers poliodystrophy. Alpers is the man in the lower, that's the lower picture. I had the pleasure of meeting Alpers' son, who is a retired professor of gastroenterology at Washington University, who looked very much like his father and came up to me after the talk and said, "I've been to thousands of lectures and no one's ever talked about my father. Where did you get his picture?" Because that's how I remember him. It was really one of the highlights of my career, just meeting Alpers' son. So the picture in the upper pictures of Peter Huntenlocker. So we talked about in 1931, Alpers describing this disorder. In 1976, Huntenlocker putting the liver features on this disorder. And then Huntenlocker said, based on the pattern of illness, he thought this was an autosomal recessive disease. Many children present with this unrelenting epilepsy, progressive neuropathy, and spasticity. That's paralysis that occurs. And Huntenlocker said that death occurs generally 1 to 20 years into the course of the disease, often from liver failure. Both Alpers and Huntenlocker were talking about childhood onset disease. Next slide. So just marching through a little bit more of the history, Dr. Bickney's at Washington University in 1992 wrote a paper coining the term Hunt-Locker variant of Alpers syndrome being associated with the fatal Depakote toxicity. Again, not saying that they were the same disease. 1995, Dr. Harding reported that Alpers-Hunt-Locker syndrome could occur in young adults. And Dr. Copeland, who gave the earlier talk, basically discovered the polymerase gamma gene. And I remember meeting Dr. Navio, who's another key component of pushing this illness forward into what we know now. I met him, I was running a conference that he spoke at, and afterwards at the conference, he asked me if I've heard about POLG and introduced me to Dr. Copeland's work. And this may have been 1997, by the way, may not have been 1996. And said to me, I remember him saying to me that he was convinced that Alpers disease was a disorder of POLG and POLG mutations. And then he went on basically changed his career to work on trying to figure it all out. The first mutations in POLG known to cause a human disease were described in 2001, causing a disease in older people with progressive ophthalmoplegia. Ophthalmoplegia means drooping eyelids and eyes that don't move to the right, they don't move to the left, they don't move up, they don't move down. The eyeballs are frozen into place. And you would say, well, that doesn't really sound like the disease we're talking about. Where's the liver failure? Where's the epilepsy? It's a very different disease. And although if you've got it, you're not particularly happy with it, you're in your 50s and 60s and 70s with these symptoms. There happens to be a higher incidence of Parkinson's syndrome and muscle weakness in people with the adult onset variety, but generally not believed to be a fatal illness. In 2004, Dr. Navio described several families with Alpers disease having distinct mutations in the polymerase gamma gene. So that was 2004. And then over the next couple of years, a bunch of papers came out, a bunch of papers in 2005 and 2006. And, I had read those papers and said, this patient, my patient case 3, must have this disorder. There was no one there's no laboratory that ran these tests, no commercial laboratory that ran POLG sequencing. And so I sent the blood to Dr. Lijun Wang at Baylor, who ran sequencing and proved that my patient, in fact, had POLG mutations and published in a 2008 publication. To complete the story, a bunch of us who were seeing these patients already talked about the fact that there must be a link with valproate toxicity and POLG mutations. And in 2010, Dr. Sinetta, who was a student of mine, had enough guts to publish a paper that was really cutting edge and controversial when he published it. But it's now a gold standard today. That 1 in 50,000 is still a number that exists, but we believe that valproate liver toxicity only occurs in people with POLG mutations. Next slide. So what illness are we talking about? So Dr. Copeland mentioned some of these, and I'm going to mention them again. Dr. Navio, I believe, put together the list of 6 distinct variants of how this disease can present. It could present with a single mutation in POLG that led to autosomal dominant external ophthalmoplegia. You could have 2 recessive mutations that could cause the same illness. Sometimes it's ophthalmoplegia, again, which is the eyes not moving up and down, left or right, droopy eyelids, can be accompanied by other features, mainly Parkinsonism, ataxia, muscle weakness, and dementia. A milder form of the disease was known as Sando, that got renamed ataxia neuropathy syndrome, and that's what my patient had. The nerves don't work, so you develop imbalance. Speech becomes slurred. You have ophthalmoplegia. You can have seizures. MRAS, which is mitochondrial recessive ataxia syndrome, Alpers-Huntenlacher syndrome, which we've described in cases 1 and 2, another childhood myocerebral hepatopathy spectrum syndrome, which is the most severe form of the disease, with horrible liver failure, lactic acidosis, and death generally around 6 months of life. But there are other syndromes that can appear to be something else, like Parkinsonism with peripheral neuropathy that can be due to POLG disease. So the spectrum of illness is quite wide. And a couple of years ago, next slide, a colleague in Norway, Dr. Hickmet, looked at several hundred cases of his that he put together of POLG disease, and decided that the best way to look at this isn't the complex naming system that Dr. Navio came up with, and Dr. Copeland and I have been writing about for the last 15 years, but breaking the disease down to before the age of 12, between the age of 12 and 40, and after the age of 40. And before the age of 12, you see the disease that has the unrelenting seizures, the muscle weakness, the feeding difficulties, and liver involvement. Between 12 and 40, a milder version that can be just as lethal, but less rapidly so. And then after the age of 40, the disease that presents generally with ophthalmoplegia and droopy eyelids, but can have some other components as well. Next slide. These slides are taken off of the paper that Dr. Copeland and I wrote. And this slide talks about the different laboratory findings you can see in POLG disease. Back before there was genetic testing, you would come up with the diagnosis based on clinical features. That's what the patient looked like, and supporting laboratory evidence, like in this case, elevated lactic acid in the blood and spinal fluid, elevated protein in the spinal fluid as well. Evidence of liver dysfunction in POLG disease may not occur for many years. We used to do muscle biopsies all the time. Up until 2010, I was doing about 50 muscle biopsies a year in my patients. Last year, I didn't do any muscle biopsies in my patients because we have replaced muscle biopsy with genetic testing. But if you do a muscle biopsy, and sometimes you back into POLG disease because you're looking for something in the muscle, you can see muscle ragged red fibers, complex 4 negative fibers, fatty deposits in the muscle as well. So next slide. Almost every patient with POLG disease will get a brain MRI scan, even if the POLG disease is not suspected. And although it doesn't project particularly well, and I don't think my pointer will work, there are certain parts of the brain that can be affected in POLG disease and will give us clues as what to tell us where to look in the genome. If we didn't, we would be looking for mitochondrial disease based on these MRI findings. Next slide. And here are the clinical features by age in patients with POLG disease. And I think that's my final slide. And maybe one more. This is another paper that I wrote with Dr. Navio that overlapped with Dr. Sonetto's paper that he wrote with Dr. Navio. So we were all writing about this like crazy 10, 15 years ago. And I want to and we're going to have some time for questions. We left some time for questions by repeating what Dr. Copeland had said about therapy. Pretzel Therapeutics is coming up with a molecule that improves POLG function. There are other companies involved. There's major international effort in trying to find treatments and cures for POLG disease. The POLG Foundation is working with the UMDF. The POLG Foundation is an independent foundation and is sponsoring a natural history study. If you happen to have POLG disease, we would love for you to participate in a natural history study that's coming up. I just got information about what this is going to look like definitively a couple days ago. So I think I'll stop now and we can open it up to questions.
Yes, and I'd like Dr. Copeland to join us on screen as well.
Thank you. Here.
Yes. There is a question in the Q&A. I'm going to paraphrase it a little bit. And this would, I think, be for Dr. Cohen. If a person has POLG and has a seizure disorder, which of the anti-seizure medications would be possible for them? Is there a list somewhere? What does the family do if they're in that situation?
So the one thing about seizures and POLG disease is you got to stay away from Depakote. I mean, that's an absolute, no drug. What we do know is phenobarbital is not particularly effective. Carbamazepine is not, phenytoin is not. We still use these drugs in some patients with POLG disease because we run out of other options. There's 25 anti-seizure medicines. Taking away Depakote, there's 24. Levetiracetam, also known as Keppra, Briviac, are probably the mainstays of therapy. I tend to use a lot of benzodiazepines, that's Valium and Valium-like drugs in my patients. But there's really way more than a dozen other medications that can be effective. Dr. Russ Sinetto has written about this, and his papers would be a good reference. I think we talk a bit about this in our paper that's online on Gene Reviews as well. So I don't want to take up the time naming names, but they're documented in the medical literature.
Thank you.
We've also used the ketogenic diet to treat some patients with terrible epilepsy due to POLG disease.
Can you tell the group a little bit about what the ketogenic diet is? 'Cause maybe people don't know what it is.
So it's basically a diet that's extremely high in fat and very low in carbohydrates and proteins. We don't know why ketosis, and it's not the ketones that are causing these seizures to get better, but we don't know what the special sauce is about the ketogenic diet that can make seizures better in mitochondrial disease. But we've used it safely, sometimes effectively, sometimes not so effectively, in treating the seizures with that are seen in POLG disorders.
Thank you. And the next question, let me see if it's still in there. Is an ATP6 deviation related to POLG?
That could possibly. So ATP6 is encoded by the mitochondrial DNA, and the mitochondrial DNA has to be copied by POLG. So it's sort of a downstream effect, possibly, but usually they're independent. They're independent diseases.
I have a
Right,
question.
but someone with POLG disease is going to probably knock out some features of ATP6 and all the other, over time, all the other mitochondrial genes.
Mm-hmm. Yeah.
How much do we know about how all of these things work together and relate? Is that something that we're going to see advancing in the future?
I'm going to let Dr. Copeland answer that because some of the research he's done gets to that.
You mean in terms of all the complexities of all these diseases?
Yes.
Well, yeah, that's sort of what I was touching on. So in terms of genetics, the whole genome or whole exome has been just wonderful in terms of getting a wealth of data, but sometimes it's actually too much. I mean, there's 6 billion copies of, in our nuclear DNA, and any one individual even within family members, there's over 1 million variations. And so trying to tease out what's the causative mutation versus the benign versus not one that's not important, that we leave that up to the bioinformatics specialist. And then tying that into environmental effects, nutritional diet, and everything else, just how people live. It's a complex question. So
Oh,
we're
for
not there
sure.
yet, but yeah.
But that's why we have people like you out there fighting the fight and trying to figure it all out. As we wrap up, we have about 4 minutes left. I have a sort of general question for both of you. In the spirit of Thanksgiving, what advancement in mitochondrial science or mitochondrial medicine are you the most thankful for, and do you think will help move us forward the most? I'll start with you, Dr. Copeland, because your face is on my screen right
Um, I would say the cost of sequencing has, we know the cost. Let me compare it to computer chips. So we know the cost of computer memory has come down, a good 1,000-fold or so. And, memory, what we used to, when you buy a computer in the 90s, now that's something 100 times that is in your cell phone. But the cost of sequencing has come down over 1 million-fold. And that has been made it more affordable for everybody to get the whole genome, whole exome sequencing. Insurance companies are even paying for this. And, what used to be costing multi-million dollars is you can sequence an individual for several hundred to $1,000. And that's probably the best benefit for just molecular diagnosis,
now. You
all
think
these
that's
diseases.
the thing that will push us forward in the future? What do you think is the next generation sequencing is going to look like? What do you think we'll learn then?
We'll be sequencing neonates and newborns very readily. But yeah.
That's pretty exciting stuff.
Yeah.
Now, Dr. Cohen, same question to you. What advancement are you thankful for, and do you think will move us forward in the future?
So I'm going to start with the same answer. I was actually at a conference, a small conference last week, where I talked about bringing sequencing into the hospital, making it, you know, making it available to everybody. The cost has come down. So our state pays for sequencing in Medicaid patients, so we can get to a faster diagnosis. Um, um, we're going to be sequencing newborns when we don't know what's wrong with them, so we can get to a faster diagnosis. This is not only going to help identify POLG patients, but patients with other diseases, many of which actually have treatments that are available now. The second thing that's coming into play is gene therapy. Actually, this got a bump forward by the pandemic because mRNA vaccines and gene therapy really are nearly the same concept. And the problem with POLG disease is that we're not trying to treat one organ. We're not trying to treat the liver, because if you treat the liver without the brain, you're seldom going to have success. Although patient 3 is a patient who got her liver treated with a liver transplant and has had quite a prolonged survival after that, even though she is sick with POLG disease. Treating the brain without treating the liver is not going to do any good. And it's hard for gene therapies to target up, at least at this stage, target different organs or more than one organ at a time. I like the question about mitochondrial transplantation or mitochondrial augmentation. There's work being done. There was a paper published by Dr. Brestoff's lab at Washington University talking about treating mice with Lee syndrome with mitochondrial transplantation / augmentation. I know that paper well because my daughter was like 8th author on the paper, so I'm very proud of her and welcoming her to the mitochondrial therapy community. She's a college student, but doing some really neat work in Dr. Brestoff's lab. So I think, there's certain diseases that could be treated with mitochondrial transplantation, although POLG is going to be a difficult haul with that therapy. So I think I'll stop there.
Margaret,
Well, thank you.
there is a question one more in the Q&A.
Is there a specific genetic panel test for POLG diseases?
There are, but I basically feel very strongly that panel testing is going to miss diseases, and it also gives you the false sense of security as you looked at everything.
I agree.
Whole genome sequencing is the best thing you can get today. It's going to be replaced by something better a year from now. So, stay tuned. I have patients that had normal whole exome testing 2 years ago, where we found the answer on whole genome testing.
Wow.
2 months ago.
Wow.
So I think we should all be thankful for the advancements in genetic testing and helping our patient families get answers that they need. Thank you both so much for this. During this, I was texting my colleagues, like, we have so many places we can put these talks that they'll benefit people, because this topic touches so many of the different disease types. I want to make a quick announcement for our guests that on December 11th, we'll have our Ask the Mito Doc webcast wrapping up 2024 with the Mito Docs. This is our ask any question as fast as you can end of the year event that we do each year. So make sure you mark your calendar for December 11th. Again, I want to do just a giant thank you to Dr. Copeland and Dr. Cohen for a really interesting talk tonight. And for all of our folks in the audience, we'll see you in December.
Thank you.
Thank you for having us.